Hi everybody, my name is Giulia Bertolin and I'm a researcher at the Institute of Genetics and Development of Rennes in France. And I'm really honored to be here with you today on World Cancer Day talking about my work. So my work focuses on the role of Aurora A at mitochondria and more specifically how it controls mitochondrial morphology and energy production. So you might know that Aurora A is considered now a hallmark of epithelial cancers, and you might well appreciate here in this histochemical micrograph, where you can observe a mammary gland tissue slice in where Aurora A is overexpressed compared to control tissues. And you can see that the overexpression of Aurora A indeed increases the size of the epithelial cells of the mammary gland, here indicated in purple, compared to a control situation. And now what we do know is that the overexpression of Aurora A results in mammary hyperplasia and in tumor formation, both in mouse and in humans. So we also know today that Aurora A is a very multifunctional protein which has a plethora of substrates, and it is highly implicated in cancer. From the enzymatic point of view, we also know that Aurora A is a serine-threonine kinase, which is active in the G2M phase of the cell cycle. So the phase right before the one that needs the cell to enter the division process. So at this step, Aurora A plays several roles. First of all, it helps the maturation of centrosomes. Here, the substrates in blue. It regulates the timing of the mitotic entry, and it helps the construction and the control of the bipolar spindle. So this structure that actually tethers away chromosomes when the cell needs to be divided. What actually we were missing was a link between Aurora A and mitochondria, at least a direct link. And why did we come up with this idea? Well, actually, as I told you before, we knew that Aurora A was overexpressed in epithelial cancers, and that epithelial cancer cells actually have a high energy demands from mitochondria. So these are rather oxidative tumors, they are non-glycolytic. And since Aurora A plays a pivotal role in epithelial cancers, we asked whether it could also play a role at mitochondria, direct or indirect. So to start tackling this question, actually we wondered first if Aurora A could have a mitochondrial localization at the subcellular compartment. And to try to answer this question, we first took advantage of high-resolution confocal microscopy. And as you might appreciate here in these immunofluorescent micrographs, Aurora A is in green, and it quite nicely colocalizes with an intrinsic mitochondrial protein, which is PMPCB here in red, which gives a quite nice yellow merge staining. And at this point, we wondered whether Aurora A was just sticking to mitochondria or if it was actually imported inside this organelle. And to do so, we needed more resolution. So we turned to electron microscopy. Here you see the representation of one single mitochondrion of HEK cells, in which Aurora A GFP is overexpressed and where it is detected with the aid of an immunogold antibodies. That gives a rather dotty staining here in black. So each dot corresponds to Aurora A, and as you might appreciate, Aurora A is indeed in the innermost compartment of mitochondria, so the mitochondrial matrix and the mitochondrial crystal. Here, there's invaginations of the membrane. This was actually confirmed by biochemical experiments. So we performed some mitochondrial extractions, here represented with letter M, compared to total fractions, which are here, T. And indeed, in mitochondrial fractions, we retrieved Aurora A, as you can observe here, and we actually could identify two isoforms, here represented in orange and in red, which are mitochondrial specific. So we were very happy because we actually observed that a fraction of Aurora A is indeed localized in mitochondria. But of course, localization doesn't mean function per se. And so to answer the second question that we asked, we first tried to detect whether Aurora A could have any enzymatic activity at mitochondria. And to again, to answer this, we took advantage of a quite nice tool that we have produced in the lab, which is a FRET conformational biosensor of Aurora A. So you might be familiar or not with FRET, but this is a rather simple concept. So it relies on two fluorophores, a green fluorophore here, GFP, which acts as a donor of energy, and a red fluorophore here, which is M-cherry, which acts as an acceptor of energy. If the two fluorophores are close enough, one to the other, we might observe the transfer of energy from the donor to the acceptor, and this is called the FRET phenomenon. And in the case of Aurora A, FRET happens when Aurora A changes its conformation, and this is due to an autophosphorylation of the protein, which is a hallmark of its activation. So what you have to keep in mind is that conformational change equals activation. This is a nice tool because we can go at a spatiotemporal resolution and analyze the activity and the activation of Aurora A directly at mitochondria. So this is how we used it. These experiments were performed in MCF7 cells, so an epithelial cancer cell line. And here you might appreciate the fluorescence labeling of our mitochondria, labeled with our biosensors, so GFP, Aurora A, and cherry. And this is compared to a donor-only construct that doesn't perform FRET at all, because it does not carry the acceptor. And the pseudo-color scale helps you going through this image, because whatever is labeled in red or in yellow indicates an inactive Aurora A, so no FRET. And whatever is labeled in blue indicates an active Aurora A here at mitochondria. So where FRET is occurring and Aurora A is active. So this is calculated by analyzing the lifetime of fluorescence. So this is one of the multiple ways that we have to analyze FRET. And what you have to keep in mind is that the decrease in lifetime equals FRET and equals the activation of Aurora A. And what was interesting in our case was that this FRET, so this activation at mitochondria, was somehow eliminated when we used MLN8237, which is roughly the only inhibitor present and available for Aurora A that now is in phase 1 of clinical trials. So this compound seems to be quite effective and removes FRET and therefore Aurora A activation also at mitochondria. Again, we were very happy. We have a localization. We have an activity of Aurora A and mitochondria, but what for? We decided to take a look at the most straightforward feature of mitochondria, so mitochondrial morphology and mitochondrial dynamics, which can be very informative in terms of functionality of mitochondria. And again, to answer this question, we turn to electron microscopy and to Hex cells. So these are this is a thin section of mitochondria in Hex cells, where organelles appear as beans, like here, or as small globules, as here. And we were very surprised to observe that when we overexpressed Aurora A, so when we created a cancer-like context by overexpressing Aurora A, mitochondria were completely and dramatically changing shape. So you can see this is one single mitochondria. Mitochondria were dramatically elongating, or they were getting swollen. So we could conclude that the overexpression of Aurora A per se elongates mitochondria, directly or indirectly. We demonstrated that this was a direct phenomenon. So Aurora A directly phosphorylates and acts on the mitochondrial fusion and fission proteins. But what we also wanted to look for was a spatiotemporal resolution of this phenomenon. So to answer this need that we had, we created a photoconversion assay, relying on Mitochondria 2. We performed this both in flies, so in Drosophila, and in cells. And today I'm presenting you the fly data. So mitochondria is essentially a green fluorescent protein that localizes at mitochondria, as shown here. And when this is quite a nice protein, because when you photoconvert it in a small discrete area, as here with a 405 laser, so with a blue laser, mitochondria from green turns to red, as here. And this is quite a visual and rapid readout of your mitochondrial connectivity, because if red remains in the photoconverted area, as in the first case here on the left, it indicates the mitochondria are not connected with each other, and therefore it infers on fusion and fission aspects of this network. Whereas if, like on the right side, you retrieve the red signal somewhere else in the cell and in the rest of the mitochondrial network, this indicates that the mitochondrial network is fused and mitochondria are connected. So this is exactly what we did in flies. So this is a wild-type fly. Before photoconversion, the mitochondrial network of an epithelial cell is just green. The photoconverted area is here in a dashed white line. And you can see that after 2 minutes following photoconversion, red mitochondria essentially remain in the photoconverted area. And again, if we overexpress Aurora A, as you can see here, after 2 minutes following photoconversion, the red signal is retrieved anywhere else in the mitochondrial network, indicated that mitochondria are really heavily fused together. So an Aurora A overexpression, even in the fly, boosts mitochondrial connectivity and fusion. Again, whoever says mitochondrial connectivity also infers on the functionality of the mitochondrial network. And this is exactly the last point I would like to cover today, is that how do we connect mitochondrial fusion and energy production from mitochondria? So you might know that mitochondrial ATP is produced within the Krebs cycle, and this is the oxidative respiratory chain, which is devoted to the production of ATP. So we basically measure the production of ATP issued from respiration cycles on this chain. And I'd like to draw your attention here on the white bars. So we compared cells that overexpress GFP towards cells that overexpressed Aurora A GFP here in white. And you might appreciate that actually in both cases, both basal and maximal respiration of mitochondria are boosted when Aurora A is overexpressed, which results in a higher ATP production rate. So the overproduction of Aurora A is not just a mere inducer of mitochondrial fusion, but it also boosts the functionality of mitochondria. So this was quite nice for a protein that actually plays a wide range of function in the cells. But what we discovered was that it also has a novel subcellular localization, which is mitochondria. And at this subcellular compartment, the protein is indeed active. It has a catalytic activity. And to do what? Basically, to modify the morphology of the mitochondrial network, inducing fusion and boosting, increasing the quantity of ATP produced by mitochondria. So this, our work, opens up the perspective, the future perspective of potentially modulating mitochondrial ATP production as a possible potential therapeutic strategy in cancer contexts that show the overexpression of Aurora A. So with this, I'd like to thank you for your attention. Please do not hesitate to contact me if you have any question or if you wish to discuss. And thank you again. Bye.